pleasure for me to be up here. Zorian joined my group in December 2019, I think was the beginning of the rotation. And I think that that probably rings a bunch of bells for people because that was a very challenging time to start a PhD. And that sort of brings me to the first thing that I want to say about Zorian is that he's been incredibly resilient. So through not only that, but through all the crazy things that a PhD throws at you. Um, and that's been projects that we thought that I was very excited about that didn't end up working out. And then lots of different new things and changes in the group and so on and so forth. The second thing I want to say about Zorian is that he's been incredibly adaptable. So he started his project working in a fully PyTorch-based environment. He'll talk about TorchDMS and switched to a new project involving sort of more mechanistic models and then moved on to like a simulation project using Java. And he'll show that all these things make sense together. But at the same time, that's just a lot of tooling and a lot of stuff to move around between. So he's done an amazing job given all these challenges. So that's sort of the end of the more serious things. He's also been just like a great human to have in the lab. And so it's always fun to look at the very beginning. So this is our first conversation on Slack. And when it says, the first thing I ask is, what is your GitHub ID? This is like the first thing I have to know about everybody. GitHub is like Facebook for scientists. And like, so I have to know what's your GitHub ID. And he's like, what's a GHID? And so, I mean, yeah, he's laughing because there's a lot that happens on GitHub in the lab. And so, okay, I'm not doing presenter mode, so I can't have the reveal. But we've also enjoyed sharing various things, including music, and other stuff. So this is actually, if you look very carefully, I noticed that this shirt says Goochland. And Goochland is where Zorian is from. It's a very small town. I mean, how many people are in Goochland?
I mean, it's growing now.
Okay.
A few 100,000 people there, but no, it's that's an exaggeration. A few thousand.
Okay. Anyway, so our YouTube history. I figure if I'm going to put Zorian's YouTube recap up here, I should put up mine. And right. So anyway, Zorian has been just such a great person to have in the lab and a lot of fun. So I'm gonna hand it off to Hugh, who has really co-advised Zorian in the lab.
Great. So for my part, I was going to note with this Slack thread that both Eric and Zorian are early risers, and that is evident through Slack. So at 4:42 a.m., Eric is posting about the stars and how he can see Venus and Jupiter. And then Zorian responds 5 minutes later that he ran outside to get a glimpse. So cool. And Eric is talking about how fascinating this is, and Zorian says, yeah, so cool. And I'm reading through this sci-fi novel series that makes him fascinated about space. And I love this, and I'm going to try to draw an analogy here. So I think like stars are one of those things where it captures like the beauty of the universe. You know, you can look up into the sky and feel wonder and amazement. With me so far? Well, I think my sense is that Zorian feels the same way about science. I think he feels this way about math, about these models that he's developed, viral evolution and things like that, and has made it really fun to work with him. Just like one example of that is a couple months ago, we were working on a paper and we were working hard to meet this deadline. We had just had a meeting that was like an hour long, and I was feeling kind of like mentally exhausted at the end. And we're standing up to go, and Zorian looks at the chalkboard, and there are all these like equations and drawings of something unrelated. And he's like, what are those? I was like, well, can I tell you about this idea I had about transformers? And he's like, yeah. And so for like the next like 20 minutes or something, he provides all these like interesting insights based on like classes he's taken and like his own research. And it was just so fun. And I think that's a really great quality. And I think it'll be like really evident in the great research that he shares today. So, Soren, want to take it from here?
Thank you so much, you and Eric. I'm actually kind of shocked you dug up so much information you want me to share, considering as my lab mates will attest, I'm never here in person, so it's probably tricky to get to know me in person, but I'm really flattered. Okay, let's get this show on the road. What's up? Yeah, it should be okay. At least when we test it. Oh, that was the first go. Yeah, yeah. Okay. Hopefully, if there are any issues with the sharing, please um, stop me before I get IT. I don't know how to do that. Let's see. Okay. How does that sound? I don't know. Better? Okay. Yeah, it's getting on my nerves too. Okay. Yeah, I'm gonna. Sorry. Okay. Okay. I'm sorry that didn't get away. Okay. Uh, where's the extra mouse? Somebody moved. Oh, here it is. There we go. All right. This is not confusing at all. All right. I think we're ready to go finally. Okay. Are we all good? Okay. Thank you everybody for taking the time out of your day to show up and attend um, my dissertation defense, which I'll be yapping for like 45 minutes about the science I've been working on for the past 5 years. Um, as was kind of preempted, I'm going to be sharing three software packages or tools that I've worked on over the years that are all kind of geared towards studying the antigenic evolution of different viruses in general. Okay. Okay. Next slide, please. Keynote's not working. I mean, I'm using both mice and my keyboard. This is let's see. Hello, IT. If you're online, we can use some assistance here.
Yeah, hey, Zori, and everything looks good on our end here.
Oh, I can't move. My screen was frozen. Let's see. Just a Mac thing? Okay, I apologize. My computer is completely frozen and breaking. That's great. Okay, let's try this again. Bro, okay. I'm using both mice. My keyboard, the black, that is my laptop. I'm clicking, nothing's working. Everything's unresponsive. Okay, I'm gonna try force quitting some things. And we're gonna stop share. Okay. Okay, yes. Okay, we have action. Well, thank you so much. It would be terrible to get failed on my birthday because of something like this. Um, anyways, okay, so I don't know. Let's get these. This is they're really testing me today. How do let's see. Okay. Well, actually, let's hide me too. Okay. Alrighty. Let's get going. I'm gonna try to make up some time, but bear with me if not. Okay, so just to kind of set the scene about what I'm going to speak about today, I would like to kind of establish some common vocabulary and give some preemptive information. So I'm going to use this term fitness landscape and terms like genotype and phenotype quite a bit during the duration of this talk. And I just want to take some time to kind of explain what those things mean. At a high level, fitness landscapes are essentially mappings between a protein sequence and its ability to perform some function or phenotype. So for those that are unfamiliar, genetic sequences such as DNA and RNA are translated into amino acid sequences, and these amino acid sequences are in turn folded into proteins. And the amino acid composition of a protein determines its effectiveness at performing some biological function. This biological function in particular, I'll kind of use the word phenotype to loosely refer to it, and how well this protein performs this phenotype. I'll use this notion of fitness to allude to the quality of the performance there. So on this cartoon example on the right, I'm showing a DNA sequence that has one mutation that so happens to actually change the amino acid at that site for these two viruses. And we see like in this amino acid mutation that I'm showing here, the fitness of this gray virus is a lot better than, or a lot higher than this magenta virus here. I'm a big video game nerd, so I use health bars for fitness, and hopefully that's okay with people. Um, just some examples of the phenotypes that I'm interested in my work are things like protein expression or the ability of a protein to reliably and properly fold on itself. Binding, in particular, since we're really interested in viruses, the ability of a virus to bind a receptor on a host cell. And then lastly, evasion from host immunity, such as like antibodies. We froze again. Okay. Animate. Okay. Alrighty. We're not frozen anymore. So this mapping from protein sequences to a quantitative phenotype is kind of what we know as a fitness landscape. So the big idea I want you to take away from this slide is that if we give a protein sequence, and we should be able to pop out some quantitative phenotype or some value that describes how well that sequence performs a specific task. Okay. So a fitness landscape in practice may look something like this, right? Where you have sequence space in your horizontal or X and Y dimensions, and then some measure of fitness or phenotype on the Y. And one thing you should take away from this is sometimes these topologies of the shape of these fitness landscapes can be very complex and very bumpy and kind of wild, right? And the reason that these topologies of these fitness landscapes are so I guess, irregular and complex, involves things that have biological impacts, such as functional constraints. So, um, essentially, when we make mutations of proteins, in most cases, they'll actually break the protein or prevent it from performing its intended function. Um, then there's also this phenomenon called epistasis, which I'll talk a little bit more about in future slides, but we can think about this as unexpected, like nonlinear interactions between mutations. And then there's also selection from host immunity. So things like T cells and antibodies actually protect from viruses by kind of targeting and binding specific sites on the protein and preventing them from making us sick. Okay. So all of these things have essentially produced a bunch of different experimental techniques and methods for studying viral evolution, but interpretable methods, especially for inferring these fitness landscapes from large quantities of data, at least when I started my PhD, were relatively lacking. And that's kind of where I step in. So for the next 40 minutes or so, I'm going to spend some time describing three projects that I worked on over the course of my PhD. The first is going to focus on inferring mutational effects and inferring fitness landscapes with Torch CMS. Um, and then I'm going to kind of take a foray away from, I'll talk about more static phenotypes with Torch CMS, and then we'll move to the world of immune escape and immunology with polyclonal. And then I'll kind of wrap things up by taking a bird's eye view of this challenge of viral antigenic evolution and provide a really cool simulator for generating nice viral populations that we can benchmark global surveillance methods for influenza against. But just to get started, I'm going to dive into my first project of my PhD, Torch CMS. So before I really get into the details, I want to spend some time acknowledging the efforts of my advisor Eric, Will DeWitt, and Jerry Galloway, who all contributed greatly with code and just helping me to become a better programmer and learning about proteins and everything like that. But okay, before I describe what Torch CMS is, I think it's important to outline the type of data outline the type of data that we're interested in and concerned with. In particular, we are interested in analyzing deep mutational scanning data. And for those that are unfamiliar, deep mutational scanning, which it's a mouthful, I'll just say DMS probably for the rest of the talk, essentially allows us to provide really large samples of a local fitness landscape of a protein. So the way this essentially works is that we essentially can create, or not we, I don't do this, but scientists in the lab will create a large library of protein variants. Here I am showing 3 different variants of some viral protein. They're all color-coded nicely. And we can sequence this library and get an idea for which mutations are present in of the variants present in the library. Alrighty. So once we have our sequences, we can essentially count them to see how many copies are present. And where the magic happens is then we impose some sort of selection on this protein library. So whatever phenotype we would like to measure or assess these proteins for, really smart people will come up for some biochemical way to kind of measure this and assess this in the lab. After imposing this selection, we can count the amount of proteins post selection, and this gives us an idea of which proteins are really good at performing this function or not. So after we tally the proteins post selection, we can actually just take the post to pre ratio of the two, and this kind of gives us an idea of like how well, with respect to the original wild type or normal sequence, each variant performs. So if we take the log of all these scores, how we can kind of interpret these scores is like if a protein variant has a negative score, we can interpret this as this protein maybe isn't as good as the wild type or the variant we find in nature at performing a function. And if the score is positive, it's perhaps a little bit better or more effective at the task. Okay. So deep mutational scanning is great, and we can actually measure hundreds of thousands of protein variants with a single experiment. However, there are some challenges that present themselves. And the one I'm going to focus on here is the phenomenon I talked about on the first slide, or maybe it's the second, epistasis. So just to kind of give an idea of what epistasis is and how it could complicate modeling efforts for these problems. Say we have 4 different viruses in the lab, and we have like this wild type or our normal, quote unquote, normal virus, and then we have 3 different variants. We have genotypes A and B, which both have perhaps a single mutation made to their sequence, and these mutations change the phenotype or the performance of the protein in their own way. And I'm showing that here on the vertical or the y-axis here. Naturally, you'd expect if we have this red variant and that has both mutations from genotypes A and B, we could just take the mutational effects together and sum them together, and everything's peachy. However, what we actually see most of the time is this doesn't match up perfectly. So genotypes A and B may check out as we'd expect, but then when we actually look at this AB genotype or this red genotype here, we notice that its value is a lot lower than what we'd expect. So in this middle plot here, I'm showing a dashed gray line over the red bar, and that's what we would expect So the thing will have to be if these two mutations added together in a linear fashion. But when we actually do the experiment, we see that this phenotype score is a lot less, and this discrepancy we refer to as epistasis. There are a bunch of different forms of epistasis. I won't go into extreme detail about them here today, but this is a big problem, especially as you expand the mutational space or how many mutations you're considering. Okay. And when we think about modeling these problems, we actually kind of want to accommodate for this phenomenon, but we want to do it in a way that's simple and doesn't add a bunch of parameters to whatever model we impose. And to do this, we use this framework or approach called global epistasis. And the big takeaway from global epistasis is that we can actually use these assumed linear or additive effects and supplement them with a simple nonlinear transformation to make up for this discrepancy. Okay, so hopefully that set the stage. And where we come in is I would like to essentially provide a tool or a software package for inferring fitness landscapes from this deep mutational scanning data while accounting for accounting for epistasis. So we have three big goals that we wanted to accomplish when we started torching this project. The first was to provide a framework that's flexible enough to accommodate for ever-increasingly complex deep mutational scanning experiments. So, for example, we perhaps maybe want to fit a model to two phenotypes jointly instead of a single model for a single phenotype at a time. We'd also like for our model to provide interpretable mutational effects. So when we look at the parameters of our model, we know which mutations are perhaps beneficial to the protein or harm the protein's function. And then lastly, as with like any general regression-based machine learning project, we would like for our model to do a good job at predicting phenotypes on variants it hasn't seen in training. Okay. So to address this first goal, I'm going to give a brief overview of TorchEMS, or the framework that we use to accomplish this task. Quite simply, TorchEMS is a neural network model that takes in protein sequences as input. So for example, we could have this protein sequence V, and it's 4 amino acids long. And then we can fit this linear model or this additive phenotype model by essentially just saying, okay, for every mutation that's possible from our sequence, we would like to have a parameter, and these parameters will essentially infer how the mutation impacts phenotype. So in the second layer, I'm essentially showing that we have, in this example, two separate phenotypes. We have expression or protein folding and binding. So we have two different phenotypes in TorchEMS. We remedy this by just fitting two separate linear models for each of the phenotypes. Okay. Once we have these linear models or these latent phenotypes in place, we simply apply a nonlinear transformation to them in the spirit of global epistasis, and we're able to actually predict phenotypes from these. The big takeaway here is that our nonlinear transformations in TorchEMS are actually arbitrarily deep neural networks instead of a more basic or bespoke nonlinear transformation. Okay. So that is essentially a high-level overview of TorchEMS, but I think the really cool thing about TorchEMS is we have a bunch of models that actually encode biophysical priors into how we infer our mutational effects. So the base case, or sort of the example I kind of just showed, was a vanilla generalized global epistasis model. This is essentially we have these two linear models, and they both share this common nonlinearity, and we try to predict phenotypes that way. We also have an independent generalized global epistasis model, and the discrepancy here, or the difference from the previous one, is instead of both latent phenotypes or both phenotypes sharing this nonlinear transformation or this neural network, we dedicate a neural network for each one individually. And oops, lastly, we have this conditional generalized global epistasis model, which is very similar to the independent model, except in this case, we're actually making the assumption that some phenotype impacts another. So for an example, we assume that like a protein isn't able to bind its anticipated receptor or target if it's not able to fold properly, right? Okay. So next we set out to assess how these models work on real DMS data. So to give these models a try, we actually went and took a deep mutational scan from who at the time a postdoc in the lab, Tyler Starr and Jesse's lab, performed a deep mutational scan of the receptor binding domain of SARS-CoV-2. For those who aren't familiar, the receptor binding domain of SARS-CoV-2 is essentially a special region of the spike protein. The spike protein, I like to think of it as like the key of SARS-CoV-2 that like unlocks the lock and allows the virus to enter your cells and start replicating. The RBD is a special region in particular. But anyways, this data set comprised over 100,000 unique variants or nucleotide variants, and on average, these variants have about 2.7 amino acids per sequence. Okay. So to train our models, we trained on about 100,000 of these sequences or 100,000 of these variants, and then we use the remaining 30,000 for validation and testing. So the really cool thing about this deep mutational scan in particular is that we actually had mutational effects reported for two different phenotypes. We had mutational effects for protein expression, and then we also had mutational effects for binding to ACE2. And ACE2 is essentially the lock on the human cells. It's the human receptor that SARS-CoV-2 has to bind to in order to enter and start replicating. Okay. So next, we wanted to take a look at how well these models did at recovering the single mutant effects reported in the publication. So here, I'm going to take some time to kind of explain how to read these plots for those who are a bit unfamiliar. But here, I'm going to show two plots. On the top in blue, we're looking at protein expression, and then on the bottom, we're looking in orange, we're looking at ACE2 binding. And essentially, what we're looking at is like, okay, if we have these published single mutant effects, how well do the Torch CMS models recover them? If the models do a great job, the points should kind of fall on this diagonal straight line. And I don't know if you can see on the screen, but this R squared value should be closer to 1 as opposed to like 0. To kind of provide a baseline or a comparison case, we fit a linear model to these data sets with no notion of epistasis or without accommodating for epistasis. And what we see here is that the linear model does, as you would expect, a not so great job at recovering mutational effects, especially for ACE2 binding. Okay. When we scale things a little bit and attach that nonlinearity in the generalized global epistasis case, we see a significant improvement with predicting expression, but actually not that great of an improvement with binding, which, if you think about it, is a kind of intuitive sense or expected considering expression and binding aren't allowed to kind of distinguish which mutations are impacting which phenotype. Oops, double animation, sorry. Okay, when we look at the independent generalized global epistasis model, we do see improvements in both phenotypes, which makes sense that the models are able to focus on both phenotypes individually. But there's still a lot of room for improvement here. And then lastly, when we look at the conditional model, this seems to do the best with inferring these mutational effects, which is encouraging because apparently our biophysical priors that we encoded worked and worked as anticipated. Okay. So that's nice. We're able to at least recover the mutational effects that were reported in the experiment, but how well do these models perform if you throw a bunch of unseen sequences at them? So that's what we're taking a look at. Here I'm showing these scatter plots again, same way you would read and understand them. Points are just colored by the number of amino acid substitutions present in any given variant. As expected, the linear model doesn't do a great job. But when we look at the generalized global epistasis model, we do start to see improvements in performance. And same can be said with the independent model. As we start to add parameters, of course, we do a little bit better of a job at predicting these things. And then, of course, the conditional model does the best. Okay, so that was really quick, but hopefully I've shown that, with TorchNS, we're actually we allow for these very flexible models to kind of be formed and trained to fit different deep mutational scanning data sets. These models tend to provide interpretable mutational single mutant effects while also accounting for global epistasis, and this is shown via accurate prediction on those held out variants. Okay. So that was Source CMS, where I kind of showed how we can use these pretty biophysically inspired neural networks to infer fitness landscapes for more static molecular phenotypes like expression and binding. But we're also interested in how mutations impact things like immune escape, and that's where I'm going to transition to now with polyclonal. Okay. Um, Once again, I want to give really special acknowledgments to Tim and Jesse, specifically with this project, and Kaylin from the Bloom Lab, and Will also for lots of regularization tips and tricks. But yeah, this is a really fun project. I'm excited to share with you all. Um, okay. So hopefully it's not too big of a stretch, given the last section, to argue that amino acid changes to antibody binding targets or epitopes of a virus can mediate immune escape. So for example, let's say we have these 5 different variants of SARS-CoV-2. They each have different mutations or varying numbers of mutations, and they're essentially distinct antigenically. If these viruses were to be challenged with some antibodies from serum from an individual, we may see something like this, where some variants are able to escape the challenge from these antibodies and others aren't. What we're really interested in with this project is how do we determine how mutations to these viral proteins, how they actually impact the antibody binding and neutralization efforts. Okay. Alrighty. Uh, Sorry, they're taking a while. Okay. Uh, Now I'd like to take some time to kind of explain what polyclonal antibodies are and what this term kind of means. So polyclonal antibodies are essentially when our immune system starts to respond to an antigen, they'll produce antibodies that bind to the antigen or the virus in different regions. So in this example here, I'm showing RBD again, and on the left side of the arrow, we have an unmutated version of RBD, right? In this unmutated state, we have 2 epitopes. We have this pink and these blue epitopes that I've colored here. And antibodies are able to bind and neutralize, or at least bind the protein at these sites. If we make a mutation to the pink epitope here, denoted by the small black X, antibodies are no longer able to bind this epitope, but they're still able to bind the blue. Uh, in the lab, we can actually measure how these mutations impact escape, and that's essentially what I'm showing here, where with escape on the vertical axis and antibody concentration on the horizontal. The dashed line is the unmutated version of the protein, and the dark line is the version of the protein with the mutations present. And what we see here is that we see a pretty significant escape from the mutated version as opposed to the unmutated version of the protein. Okay. So what would happen if we made multiple mutations in the same pink epitope, right? We'd probably expect a little bit more escape because we're just mutating the heck out of the epitope, right? In general, we may not see that big of a difference from the previous case where we just make a single mutation to the epitope. And a big reason for that is because, at least the way I like to think about this is, um, you've already kind of obliterated the ability of the antibodies to bind to that epitope with the first mutation. So you really see diminishing returns when you make the second one in most cases. But if we consider the case where we make a single mutation to both epitopes, we see the both pink and the blue epitopes. We see something like this, where um, we see dramatic or significantly more escape as opposed to the first 2 scenarios. Okay, so basically, the big point there is you just can't get away with like counting mutations to infer escape. There's a little bit more nuance to it. And our goal here is we would like to infer mutational effects from polyclonal serum at the epitope level. So in practice, we may obtain some data set from the lab like this, where we're measuring escape from antibodies from a viral protein of interest. And we'll have like on the horizontal axis, we'll have amino acid site, and then escape on the vertical axis. And the peaks here essentially denote sites where escape is happening when these sites are mutated. One thing I found out from you all are sometimes these viral proteins can be hundreds of amino acids long. Epitopes aren't that long, so obviously we see like these 3 peaks. There are probably 3 different epitopes that are being targeted. We don't know these epitopes or these regions a priori, right? And this is essentially the goal of polyclonal is to resolve these and also provide accurate mutational effects. Okay, so how do we do this? We cleverly, we have this really neat tool called polyclonal, which is very similar to TMS, but is a slightly different. So essentially, polyclonal is another model that takes in protein sequences as inputs and learns this linear model for mutational effects. Unlike TORCH-CMS that focuses on learning mutational effects for phenotypes like expression and binding, polyclonal is attempting to learn mutational effects that impact things like ultimately escape from an epitope. So if you make certain mutations, um, are antibodies that target certain epitopes still able to bind them? Once we kind of have an idea for how mutations impact escape at these specific epitopes, we can essentially combine them all together to see if this variant would escape from the challenging serum overall. And just two assumptions about this model is the first is, of course, this linear model I explained earlier, the mutations have an additive effect. And then lastly, we're assuming that antibodies don't compete with one another to bind to like the same epitope. Okay. Um, so to validate this model, we turned the simulation. We simulated two deep mutational scanning libraries with 30,000 variants each, with three real life epitopes that have distinct measured mutational effects. So that's what I'm depicting here. We have RBD once again, but this time I'm showing three panels where we're coloring our three different epitopes with the colors blue, pink, and green respectively. When we take a look at the simulated escape profile, it may look something like this. And this is essentially what we're trying to recover. Okay. When it comes to assessing how well polyclonal will recover these parameters, we essentially trained the model and compared the inferred parameters to the true simulated ones. And here I'm showing a scatter plot of the results. Each panel is essentially the mutational effects inferred for every epitope. And we see we do a pretty good job, especially on epitopes 2 and 3. And we consider the auto-sample prediction task. We take our simulated test set and we can predict escape at a certain antibody concentration. And we also see that polyclonal does a really great job at predicting escape on those variants. Okay. So hopefully, so now we're able to take with polyclonal, we're able to take these escape profiles from deep mutational scans. And in the past, these epitopes were pretty tricky to resolve, but with polyclonal, we are able to resolve them pretty effectively. Okay. All right. So that kind of concludes the I guess machine learning component of the talk. And now I'm going to switch gears and talk about the simulation tool that Eric alluded to earlier. Okay. So before I get started on this, I want to also give a special shout out to Hugh, who I worked with for a very long time on this project, and Tian, who's a very talented undergrad in the lab that helped a lot with the Java as well. And then John, Marlon, and Trevor, of course, for being our wise oracles and our guiding lights during the application phases of this project. Okay. So we're going to switch gears a little bit. I've been talking about COVID for the past two parts of the talk, but now we're going to switch gears to a virus that evolves even faster in influenza or H3N2 or seasonal influenza. So as most people know, flu evolves really fast antigenically. And this is why we have to get our flu shots like every year. Or you're supposed to get your flu shot like roughly every year. But because of this, there are really strong or really prevalent global surveillance efforts that take place to monitor and kind of predict how flu is going to evolve and to predict how we would like to craft our vaccines in the future. Two key components or two key questions that these surveillance efforts sort of ask is what kind of viruses are circulating currently? So here I'm showing a tree from NextStrain where we have a bunch of sample flu sequences that have been characterized, and we can gain an idea of like, okay, what's circulating and how fast are these populations of viruses growing? And then another thing we're also interested in is like how antigenically similar are current viruses to past viruses. Um, and this is something that is typically done in the lab, right? And I won't go into extreme detail about how these experiments work, But if we were to sample a bunch of different flu strains from a bunch of different seasons and challenge them with sera that was raised against them, we can obtain an idea of like, okay, what neutralizes what, and how similar are these viruses to one another in the context of how our immune systems recognize them. Um, and so we're able to take this data and a nice conceptual mapping, or I guess the fitness landscape version of this problem is something we call antigenic space. So we're able to take these viruses and kind of project them or plot them in this Euclidean space in such a way that antigenically similar viruses will land closer to one another. Okay. So we have all this data and all these really cool ways to characterize them, but there's still some work to be done. For example, we have a bunch of these sequences, and the first task that we actually want to tackle is grouping them into antigenically similar groups, or organizing this information from these genetic sequences into groups that look something like this. So essentially, we want to be able to take just the sequences of these viruses and assign them to groups such that these groups kind of trigger the immune system in similar ways. And this is kind of the cartoon I'm depicting here, where these viruses have been grouped into these three separate variants, this blue, yellow, and red group, and they all invoke different responses from the immune system. Um, you could do this in the lab, kind of like I alluded to on the last slide, but it's not very practical to do it for every single sequence we sample in nature, and it's also rather expensive. So it's really common and quite practical to turn to computational approaches. And I'm only going to talk about two in particular, and I'm not going to go into detail on how they work necessarily, but um, two common methods we use. The first is like sequence similarity. We can look at how similar sequences are in our sample and group them according to that metric. And then we can also take these sequences and infer a phylogenetic tree and perform analyses and algorithms on this tree to suggest clades or variants as well. Okay. Once we've organized all the sequence information into antigenic variants, the next step that researchers typically take is to take mathematical models and use them to try to predict which variants are going to circulate in an upcoming flu season. So we may have an example like this where we have three different groups of flu variants in circulation, and we would like to predict in 1 year in the future how these variants will kind of like grow or expand as a proxy for like how fit they are. There are a bunch of different ways to kind of make these predictions, but I'm going to focus on this one in particular. Um, and we're focused on these growth rate models that our collaborator Marlon devised, and they essentially take in surveillance data, like sequence count data, and use that information to predict frequencies and fitness going forward. Okay, so those are really two critical components or two critical questions that we have when we're thinking about global surveillance and forecasting and vaccine development efforts with influenza. However, like we thought it'd be really cool, okay, if we have a simulation tool to generate data sets that allow for like benchmarking these methods and actually seeing how well they work and what their strengths and weaknesses are. And that's essentially what we set out to do with this project here. So we have three big goals I'm going to kind of briefly touch on in the next few minutes. The first is our method for simulating ground truth viral populations. So we would like to simulate viral populations that have both genetic sequences and then some notion of an antigenic phenotype. And then we'd also like to be able to assign ground truth fitness values for these viruses. And with this information or with this data, we would then like to benchmark those two variant assignment methods I spoke about on the last slide, and as well as those growth rate or growth advantage models that I spoke about. Okay. Alrighty. So for the first point, or the simulator in general, we developed Antigen Prime, which allows for like joint simulation of the genetic and antigenic evolution of viruses. And the way it works is pretty simple. So viruses are actually parameterized as a 2D point in a two-dimensional Euclidean space, and we essentially attach genetic sequences to them. And as these viruses mutate, we update the sequences and move them in the antigenic space accordingly. The magic antigen prime is essentially how we move the viruses. So essentially, we choose a site to mutate at random, and then we determine how far the virus is going to move in the antigenic space depending on the type of mutation that has occurred. Okay. So first, there's like no amino acid change, right? No amino acid change to the protein in general. The protein is going to kind of stay put, right? The immune system isn't, it does the protein itself won't look much different to the host population. If an amino acid change does happen as a result of this mutation, we then have another step in the decision tree to take. If the mutation happens at a non-epitope site, we'll actually draw the step size for this mutation from a distribution that looks something like this, where it's essentially going to be a really small distribution that kind of keeps the virus kind of in the same place. It's not really going to move very much. The big idea here is like mutations to non-epitope sites don't really change the the antigenic profile of the virus very much. In the case that amino acid change happens in an epitope site, however, we're actually going to draw a step size from a distribution that looks more like this, or the virus is going to be able to move further away from its parent or the viral population. Okay. Antigen also has individual hosts that are able to get sick and recover and go back to the population and accumulate infections over time, and uh, essentially drive the antigenic evolution of these viruses. Individual hosts essentially just have their own immune memory, where they essentially store the coordinates of viruses that infected them in the past. All right, so for this host here, they've been infected with 3 different viruses over the course of their lifetime. And if you plot them in the antigenic space, they may look something like this, right? Then this new potentially infecting gray virus comes along and tries to make our host sick. The way we determine if an infection occurs is by looking at the Euclidean distance between this gray virus and the closest thing in the history, right? So, for instance, this yellow virus is really close, and we can calculate this distance and pass it into this infection risk function that essentially pops out a probability of infection. And in this example, we get a really small probability. And the big takeaway here is that these antigenically similar viruses and a host immune history provide protection from new strains or new variants. Okay, so that's a very high-level, quick overview of like how antigen prime roughly works. So in order to benchmark these variant assignment methods and forecasting models, we simulated 30 years of flu-like evolution. We had 3 demes, or you can think of these demes as 3 separate countries in different parts of the world that each have about 30 million hosts each, and then we sample roughly 150,000 viruses and also weekly case counts. And the resulting simulation looks something like this. Here's a phylogenetic tree of that was inferred from the sampled sequences, and we see that it does exhibit some of the trademark flu-like ladder-like structure that we see in nature. And then here's the antigenic space that we can use for especially benchmarking those variant assignment methods. Okay, so we have our ground truth populations. So like now what? What do we do with them? Alrighty. So on the front of comparing variant assignment methods using antigen prime, we took a pretty simple approach here. So we have always sample viruses, and we also have these hosts or their immune histories. So we're able to calculate fitness for all these viruses. And when we're able to do that, we're also able to like calculate or assign a variant to all these sampled viruses via k-means clustering on the antigenic space. Right. So this is going to serve as our ground truth. If we just kind of perform k-means clustering on the antigenic phenotypes for all of our sample viruses, we can actually get really compact antigenic clusters in this regard. Okay. So we have our ground truth, and next we wanted to essentially compare how well the sequence-based and phylogenetic-based methods do by comparison. So essentially, I ran both of these algorithms to infer roughly the same amount of clades for both methods. And just kind of qualitatively here, you can see that, okay, they're picking up on a lot of the same patterns, which is encouraging. Okay. Qualitative checks are one thing. So the next thing we want to do is to make sure, okay, well, are these variant assignment methods finding patterns that we often observe in nature? So we had two kind of criteria or goals that we use to judge these methods. The first being, are these methods able to successfully partition the sequences in such a way that there are about three distinct variants that circulate in any given year? And here's a plot that shows that all these methods do a fairly good job at this. I'm showing time and years on the horizontal axis here, and the number of distinct variants that were found circulating at any given year by each method on the vertical axis. And while there's a lot of variation and bumps going up and down, for the most part, they hover around this red dashed line here. Next, we wanted to see, okay, we have these variant groupings, but are the groupings that these algorithms finding, are they actually grouping antigenically similar variants together? Um, and to do that, we essentially calculated the fitness for every virus in our simulation over time, and calculated the variance of the fitness for all of our inferred variants during the duration of the simulation. So here is a plot I'm showing the average variance found in fitness of the variants. That's a mouthful. But the big takeaway here is the lower the line is the more compact or more similar the sampled viruses and each variant group are to one another. So we would like ideally a pretty low line here, which we see with our blue ground truth, antigenic ground truth, and then the orange and the green. It looks like they're doing a bad job, but the scaling here is a little bit deceiving, but they also do a pretty good job at finding pretty antigenically distinct uh, variants, which is really impressive because they have no idea of the antigenic coordinates and antigenic phenotype. So they do a pretty good job of just looking at the sequence and the phylogenetic tree structure alone. Okay. So that's nice. We've kind of like given a nice check to these variant assignment methods, and lastly, we want to look at how well these growth advantage models do, and particularly how well they do at inferring growth rates instead of like frequencies. Okay. Um, just to kind of explain like what's going on with these models. These models take in two inputs for the most part. They take in weekly case counts that we also get from Antigen Prime. We have a weekly surveillance of case counts in each country that we're able to provide to the models. We also have variant frequencies. So here I'm showing in this example, the dots are like the ground truth observed variant frequencies from the simulation. And then these lines are just the median of the posterior that the model inferred or the model predictions. Okay. We can take those two first panels and then we can calculate variant incidence. But I like to think of it as like, okay, if we have X amount of cases, how many can we attribute to this blue variant and how many can we attribute to the orange variant? All right, and we can then take the derivative of variant incidence to achieve these rough estimates of growth rates here. And in this last, this is actually what we're really focused on in this section. Assume like can we accurately infer how quickly each variant is growing? Once again, the dots here are the ground truth or empirical growth rate estimates. And then you can't see the line because the models are doing a really great job. But there's a line here that the models are predicting, and they're just kind of hitting under the points there. Okay. So for the most part, this section, so for the most part, these models do a really great job. It's kind of boring. For the most part, they look like this, where they're doing a really good job at inferring the variant frequencies, and they also infer growth rates relatively pretty accurately. However, one thing we noticed when we were looking at some of the worst performing windows is that there are these weird or sort of quirky phenomenons where the models tend to like overestimate the growth rates, especially when there's a surge in the case counts near the end of a training window. So in this example, we have a nice little bump in case counts that kind of aligns with the overestimation of growth rates for especially the green variant, but also the purple and red are also pretty biased. And then we also have these other two examples in their own way where like, and they all are experiencing their own surges in case counts. But even though frequency is predicted relatively accurately, we see this really big bias or overestimation in growth rates, which um, if you ask me like why this is, like I couldn't tell you, but it's a previously unknown finding that we were excited to kind of pass along to Marlin and Co. But yeah, that was a really interesting finding. It was really nice considering like most of these models were really great jobs. Like, okay, we didn't waste our time. We found something interesting. But okay, so I've kind of tried to speed run this given the circumstances, but I've briefly kind of walked through three tools that I've worked on over the course of my PhD. The first being TorCMS, which is a really flexible tool for inferring fitness landscapes and mutational effects from deep mutational scanning data. Then there's polyclonal. I talked about polyclonal, which is very similar in spirit, but focuses more on immune escape, especially from um, epitopes or multiple epitopes in polyclonal serum. And then we took a bird's eye view and we looked at things from the population level, and we kind of looked at the coevolution of viruses and host immunity. And then we showed that we can use these really cool simulations from Antigen Prime to kind of benchmark or give a little taste of what you could do with it. And you can benchmark like these really crucial global surveillance methods for influenza. Okay, I think I actually finished on time. Wow. And with that being said, I want to just give a really big shout out and acknowledgement to the NASA group. If you're like me and you've been in the lab for like 5 years or 6 years now, there's a long, I've overlapped with a lot of people. I tried to put everybody's name here. I apologize if I've omitted any. And then I also want to give a special shout out to the collaborators I worked with during a lot of these projects. So especially out of lab. So Tyler with TorchEMS and my fellow Boston Celtics fan, Tim Yu with Polyclonal, and as well as Kaylin with Polyclonal as well. And then John and Marlon, who were invaluable with Antigen Prime. And then also my committee members, Jesse, Ivana, thank you so much again, and uh, Eric, Kelly, and then I also want to thank Sarah and Tim, who couldn't be here today, but they also served a really vital part in my time here at UW, and I'm really thankful to have met them and worked with them. And then, of course, GS admin and Fred Hutch admin in IT, especially Brian and Ruby. I feel like at least with GS, I know things would be a bit of a mess without Brian, and the same here can be said with Ruby and the comp bio department. Um, but yeah, I guess now I will open the floor for any questions or comments. Yeah. I don't know how this is supposed to work, but do we want to do in person first or Zoom or Jesse? Yeah.
So, I mean, I used to wait 30 years, something like that, of evolution, and you were later talking about how you were thinking that humans did different variants, so it was like 6 or 12 months window and taking them 6 or 12 months. Was that right?
Yes, 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 yes.
So, and what performance do you envision the future rather than depending on prior 6 or 12 months, than on prior years, 4 years, 6 years? And what happened to you?
Yeah, that's a good question. So for those who maybe didn't hear, just there are also microphones. I probably should go with that. If people want to ask a question, there are microphones. But I'll repeat the question. So Jesse essentially is asking, we simulated 30 years of flu evolution for Antigen Prime, and we trained on, I didn't mention this, but we trained on like a year of data. And Jesse's curious about, please correct me if I'm wrong, if do you see anything different, or do things change if you like change the training window? If you maybe train on like 2 or 3 years of evolution, and that's a good question that I didn't really try out much with Antigen Prime. I didn't try to extend further out. I actually tried to shorten the time frame. So instead of a year, I tried like 6 months, 3 months, and performance at least when you get really close tended to get pretty bad. And I know I think John recently actually had a paper where they studied like performance of these models with different like lag times, which is a bit different. But to answer your question, in terms of like forecasting and predicting, we didn't really focus on that. So I didn't really get a good look. We kind of just focused on the growth rates themselves, like at present. Um, and when we modulate the training span, those bumps and deviances still kind of show up near the end. So it's consistent as long as there's like a surge at the end. It seems to kind of bias things, but yeah. I think John had a question.
Model predictions got better and better, but there's always this cluster of points that are on the map where the observed point is just like really low score, but then your model could have just have like a normal distribution around the possible values. Does that give a sense of like what was going on with those particular data points? Because there's something you can model, or is it just like reflecting that those are truly out of sample?
That is a great question. So, okay, for once again, I'll upload the slide. So John's essentially asking about EMS and essentially how like there's this big blob of points. We used to call this so back this I used to, so it's been like 4 or 5 years since we were working on Torch EMS, but we used to call this the wall. And it's we I don't know, the models just apparently does not do a good job near the lower threshold or lower bound. And I don't have an explanation as to like why the models struggled at those low like performing variants, honestly. But yeah, it's actually a big reason why especially Best kind of got derailed because we couldn't figure out like how to solve this wall problem. But yeah. Jonathan, yeah.
Yeah, a question about your Genome Prime paper. So when you do this amines clustering in the antigenic space, how do you pick the number K?
That's a great question. How do we pick the number? So Don is asking with antigen prime, when we work through these random assignment methods, how do we choose K? And the brutally honest answer is it was just kind of a heuristic, like, oh, we have 30 years, we should have one dominant variant a year type of thing. We did have, and this is probably why I'm giving this talk in March and not like sometime in 2025, we had a nice fiasco where we tried like more how do I say, like methodical ways of choosing K, like more heuristics that are generally used in machine learning. And with the nature of like the antigen prime data, because especially since these pictures is like a straight line, we'd only get like 6 or 7 clusters, which, you know, kind of it defeats like the purpose of like the influence and motivation where flu evolves really rapidly antigenically. We want to make sure we have like adequate turnover for variants for the forecasting models. So it was like a balancing act of like that's kind of what we'd expect in nature, but also for the forecasting task or the growth rate models task, we wanted enough variance present so we could actually like train the models. But yeah, it's a good question. Okay. Uh, Anybody on Zoom? I guess I don't know if people unmute themselves or whatnot, but okay. Oh,
I was curious. So it sounds like basically before you sort of assume this like linearity of these relationships between these like mutations and the results, right? And the contribution is like you switch out the linear model for a neural network, right? Which is like basically a nonlinear fit. So I was curious though, like it seems like the nonlinearity is really important for this contribution. What was the nonlinear function that you picked? Is it like ReLU or
oh yeah.
like log something, or was there some like physical motivation for that, or did you choose some machine learning canonical function, or what was it, the nonlinear transformation?
Cool. So Jonathan is asking about Torch CMS and essentially how we went about choosing the G function of nonlinear transformation. And that's a good question. So I didn't mention this, but well, maybe I alluded to it. But in the original like global epistasis framework, the whole idea is we can fit a linear model to use mutational effects, and then to accommodate for like the discrepancy that we see in the actual DMS scores, we can transform that linear model with some kind of nonlinear transformation. I believe they use like monotonic I splines in the original work. We went with neural networks, and truth be told, like G or the neural network we chose was kind of chosen by like trial and error. So like essentially doing like hyperparameter search where we just give a bunch of different architectures and train them all. And whatever ones, whatever architecture seemed to perform the best, especially with uh, like without overfitting, we just kind of went with. So for these models, so okay, for the GGE model, there's only one neural network, right? They share a nonlinear function. So one thing to note is like I tried to accommodate for the number of parameters between this and then the other two models since they had their own. So like the IgGE and the conditional GGE model, both actually, they all actually have really narrow neural networks. Like we're talking like 2 layers, 10 neurons, ReLU activation. But for the GGE model to accommodate for like the parameter discrepancy, I just essentially match the nonlinearities in size. But yeah, it's essentially just like kind of brute force heuristic search to discover those architectures. Yeah. Okay. Any other questions about science stuff? Okay. No. Sorry, the big technical fiascos, technical thing made me shut down Keynote and I had to put re-release slides up. Okay. Okay. Now, here, I just want to, oops, some personal acknowledgments and shout outs. First to Eric and the rest of the Matson group. So I've been in Eric's lab for a very long time, and I genuinely, whenever people ask me like, how is it like working with Eric, I always say he's one, the kindest person I've ever met. Like if people have met with Eric, he's a genuine joy and a pleasure to work with. But he's also been a really great mentor, and I feel like I've grown a lot as like a scientist and a software engineer. And I'm really appreciative for like being able to spend so much time with Eric and work with him over the years. I apologize for the people that are in attendance. You may not be in these old Matson group photos from like the COVID days. And the vegan Thai place a few weeks ago, but it's been a really pleasure working with all of you as well. So yeah, a big special thank you to Eric. And I'd also want to note, I remember in the spirit of, this is unscripted, but in the spirit of the Slack messages, I remember when I first joined the lab, Eric asked me like what I wanted to get out of my dissertation or my PhD, and I didn't really have any big scientific goals at the time. I just kind of said I want to become a better software engineer. I want to be able to write production of code, which shout out ChatGPT and Claude, I guess now. Um, and then I said I really want to learn about, deep learning. And as like coming from like a stat background that didn't really dabble too much in machine learning, that was one thing I really wanted to indulge in. And I'm really grateful that Eric was able to find projects that allowed me to do all of these things, even though they kind of felt scatterbrained and all over the place at the time. They all grouped together really nicely at the end. And yeah, thank you for the great opportunity to kind of grow as a scientist and a person. I want to give special shout outs to Will and Hugh, who also served as like really critical mentors during my PhD. Will DeWitt more in the early stages when I was kind of scared to ask questions and share ideas because I thought Eric would yell at me and tell me I was stupid and I had to leave the lab. So I'd relay them to Will, and he would be like, "Oh, that's good," or "That's not so good," and we should think about that a little bit more. And I appreciate that because I learned how to think about these problems and essentially build a bit more of my scientific confidence over the early years of my PhD. And Hugh has been great. We've worked together for like I think 4 years now, however long Antigen Prime took, and it's been a pleasure. I mean, from you know, I guess stressing over how difficult it was to produce like a flu-like simulation with Antigen Prime to then trying 4 or 5 different applications that seemed useful and worked. The patients and just the meetings were always so fun. And yeah, I definitely learned a lot about like proteins and protein engineering and viruses from Hugh. And it's always fun. And we always every meeting we always close talking about like soccer. He's a big Liverpool fan, and I'm a Barcelona guy. And I'm really gonna miss those chats about like battling and how well we're all doing in the Champions League, respectively. So it's really fun. I want to give a really quick to my undergrad advisors, specifically like Dr. Jane Roberts-Navia, who was my academic advisor at Virginia Tech, and really kind of motivated me to even like dip my toes in the research space, and specifically science. She's been super supportive and just a really great friend, even since I've graduated from Virginia Tech. And yeah, I'm really grateful for our relationship and friendship. Um, also want to thanks, Shane. I also want to give a special shout out to Dr. Jim Bruce, who is actually like my first experience working in a lab, scientific lab here at GS. Ironically enough, I worked on writing some software to study protein-protein interactions, which little did I know that would be kind of in the spirit of what I would do in the future. But Jim was great, especially welcoming in a person who knew nothing about genetics or proteins or cross-linking or mass spectrometry. It was a really great time, and it definitely played a part in me coming back for grad school. And then I want to thank my two undergrad research advisors, Dr. Leah Johnson and Allison Tagey. Over at VT Stats, I spent most of my time in Dr. Johnson's lab, working on epidemiology problems and like blue tongue virus. And um, she really introduced me to like epidemiology and Bayesian statistics in general. So those are some really great time spent in her lab. And then Dr. Allison Tagey, I spent less time with. It was only a senior year thing, but I like to give her credit for giving me a crash course in computational biology and the grad school life. Like with Dr. Taggy, I was able to experience a bunch of different conferences and actually get my hands on RNA sequencing data, which in the VT stats department, it's very hard to find biological problems to work on. So it was really fun to work with Dr. Taggy and essentially decide like, okay, I do want to maybe go into the science space and not just um, sell insurance or something like that with my stats degree. Um, Next, really big special shout out to my family and especially for these next few slides. There's no photo or name. Please chalk it up to time and space and not my mind or heart. But my mom and sister made the trip out, and then of course my parents, my aunties and uncles, they're all on Zoom, cousins. Without you all, I would literally go insane. And living 2,000 miles from each other is rough. But I'm really grateful that we all stay in touch and the group chats are will keep me going when things get tough. Then I want to give a special shout out to like my fellow GS cohort. Most of them are gone now. But and then also just my friends that I've made in genome sciences in general. Some of you are here, so thank you for coming. But I love you all, and you've made this experience, especially during COVID, really enjoyable and worthwhile. Then I have friends, and here's some personality for, I guess, my lab and advisor and everything. Um, these people are mostly people from childhood and college. They all mean a great deal to me. And yeah, I hope they're all watching, and I'm proud of them all in their own way. And yeah. And then for my Seattle friends slash my gym family, who comprises like 60% of the crowd right now. Y'all are awesome, and probably the only reason I'm like trying to stay in Seattle for a little bit longer. But I love being able to like yap at you all at like 5 or 6 a.m. in the morning, and you don't hate me too much. Um, I don't have any pictures of Stephen, but Stephen is also here as well. But yeah, I just really enjoy being able to see you all every morning and having a reliable and strong, steady friend group that I can kind of rely on when things get rough. And when things are really great, they are going to force me to celebrate, even though I'm not big on that myself. But yeah, big shout out to everybody here and everybody that's not pictured. I just want to make a comment on the bottom right. My friend Q, my friends like to pick at me, and Q thought it was a good idea to dress up as me for Halloween one year at the gym. So here that picture is there. Um, but yeah, that's all I really had to share. And yeah, thank you everybody for coming. And I don't know how this works. I think people go outside and then my committee will grill me for a while and tell me if I pass or fail. And I don't know if there are snacks, but yeah, thanks.